Alright, so welcome back to the next session of meiotic division 2. So we've been talking about cell division, which will divide into mitotic and the meiotic phase. We've talked about the mitotic phase and we're looking at the meiotic phase now. We've laid the background knowledge that meiosis occurs in two stages. There's a reduction division. Reduction division in which a diploid cell is being reduced to an haploid cell. We've seen what a diploid cell is. A diploid cell is a 2N cell, and why an haploid cell is an N cell. So, from our first stage of meiotic division, which occurs from our prophase to telophase, we are starting the second stage now. I want you to remember that from our last stage, we have two daughter cells having each chromosomes. So we have the first stage now to the prophase 2. Prophase 2 stage. We have the prophase 2 stage of meiosis, and like I'll always say, let's give a pictorial view so that you can just get a glimpse of what is going on. Alright, so these are the two daughter cells that are formed so this is a representation. So we have the two daughter cells in the prophase 2 division. We can see what happens at this prophase 2 is quite much more similar to the ones that happen in the prophase stage of mitotic division. What you have is just, you have this chromosome condensing into a thicker structure. You can see them, they are more visible in each cell. They condense into a thicker structure. The chromosome of each cell, they have a thicker structure. So this is just what you see when you observe a cell that is undergoing a meiotic division. For example, a sperm cell or an ovum. So when we observe the prophase 2, you can see that each daughter cell is having its own chromosome. So the prophase 2, you can see we have two daughter cells now, and you can see there is also centrioles. You have formation of centrioles. At any prophase stage, you have formation of centrioles. Centrioles start to form, and during the late prophase, what you just notice is that the centrioles start to move apart and spindle fiber starts to form. You've known what the spindle fiber looks like. They're just like rope that allows the chromosomes to move, or chromosomes or the chromatid to move apart. So these are centrioles. This cube-like structure are called centrioles. They are the one that develops in the spindle fibers. So this is the nuclear membrane. So each cell now undergoes what we know as division. So we have a division on the first cell. Let me just say this is the first daughter cell, and this is the second daughter cell. So each daughter cell now undergoes division. So at the prophase stage, what happened is just similar to what happened at the mitotic prophase. So let's move on to our next stage from prophase. So from our prophase 2 stage, we have what we call metaphase. So we have what we call metaphase 2. So metaphase 2 also occurs to each daughter cell. We have two daughter cells now having their own unique chromosome. So you can see this, let's just say this is the first daughter cell with its cell membrane, and what happens in any metaphase is that we have the centrioles having moved apart. Metaphase 2, you have the centrioles moving apart, and there is formation of spindle fibers. So the characteristics of any metaphase division is that there is formation of the spindle fibers and the centrioles have moved apart. When the centrioles has moved apart totally, we have the formation of spindle fibers in which the chromosomes are arranged on. So you should also take note that the nucleus and its nuclear membrane has so the nucleus has dissolved. So the nucleus has dissolved and you have the formation of a spindle-like fiber. So what happens is this. So you can see the chromosomes in each daughter cell. The second daughter cell is here. The chromosome in each daughter cell starts to separate. So this is spindle fiber for the second cell also. So you can see let's just say we have the chromosome. So what we have here is that we have in the metaphase formation of spindle fibers, and in the metaphase the chromosomes are arranged on the equatorial plates. They've not started to separate. They are arranged on the equatorial plates first before they start to separate. And at the point of separation, that's where we move to our anaphase. At the point of separation, but the chromosomes now, they just align at what we know as the center of the equatorial plate. You can just see the two daughter cells, the first one and the second one. That's the first and that's the second, both undergoing metaphase. So we have the alignment of the chromosomes on the spindle fibers. You can see the spindle fibers and you can see the chromosome. The chromosome aligned at the equatorial plates of the spindle fibers. So the next two phases, which is anaphase and telophase, will be discussed in the next video.